what we have developed are non-surgical brain implants. More than 3 billion people around the world suffer from different neural conditions, and existing implants can only help less than 1% of the patients because of the surgery-related costs and invasiveness. So by completely defying the need for surgery, we are breaking the barriers of age, health risks, and costs, which are currently preventing millions from getting treatments.
My name is Eva Cornman. I'm an educator here at the Museum of Science, and today I sat down with Deblina Sarkar, founder and director of the Nano-Cybernetic Biotrek Research Lab at MIT. We spoke about her innovative research that could one day change our approach to brain surgery. You have been working on something so crazy that it has been likened to science fiction, and I'm talking about your surgery-free brain implants. What are these and how do they work?
So, according to World Health Organization, 3 billion people around the world suffer from different neurological conditions. So be it Alzheimer's, Parkinson's, mental illnesses like schizophrenia. But currently, most of these diseases are untreatable through drugs. So though it is known that an implant which can provide precise electrical stimulation is a very promising modality, existing implants require a very invasive surgery that requires creating a hole in the skull and putting a centimeter scale probe in. Highly invasive, costs hundreds and thousands of dollars to do such kind of surgeries. And because of the invasiveness, such implants are only limited to less than 1% of the patients. So to overcome these challenges, what we have developed are non-surgical brain implants. So these are tiny electronic devices which can be introduced through the body fluids, and they have the capability of traveling through the body fluids, and they autonomously recognize a target region in the brain. By autonomous recognition, I mean they do not require external guidance or imaging to locate the target or disease region. That recognition is done through biochemical sensing. And once these tiny nano-electronic devices, they detect and recognize the targeted disease region, they actually cross the blood-brain barrier. So this is the first demonstration of electronics which can cross even intact blood-brain barrier, and they self-implant in that brain region. And once they get self-implanted, we wirelessly power them through electromagnetic fields, so they can treat diseases which are even resistible to drugs. Or they can treat diseases where drugs are ineffective.
Can you explain what the blood-brain barrier is and just how small do these implants have to be able to cross that barrier?
Blood-brain barrier you can think of as a protective film. So basically, because brain is, you can think of probably the most important organ, right? Brain defines what we are. So the brain is normally protected by our body. So if you were to inject something inside the body, it will go everywhere, but it does not enter the brain. Because that's the mechanism with which the brain is protected, so that some external things do not get inside. But this protective layer, which is called this blood-brain barrier, also prevents useful treatment-related things from getting inside the brain. So it's very challenging to get anything inside the brain. And small molecules are still possible, but if we are talking about electronic devices, that is almost like impossible to get in. So the way we have made this possible, this impossible thing possible, is because we have conjugated our electronic devices with a biomolecular machinery. So this biomolecular machinery, what it does, it gives some signals to the blood-brain barrier so that it opens up a little bit and it can squeeze through intact blood-brain barrier and enter the brain tissue. So the electronic device's functionality is to harness the electromagnetic waves and power the electronics to treat the diseases through electric fields. But crossing the blood-brain barrier and recognition of the target region, that part is done by the biomolecular machinery. So this is a hybrid of a biomolecular machinery and an electronic device. So just making the device small will actually not help in get inside. It has to have this active mechanism for crossing the blood-brain barrier.
Mm. So just how small is it, and how do you make something that small and then couple it with the biomolecular machinery that is so essential for crossing the blood-brain barrier?
Mhm. So the devices that we make are in the nanofabrication facilities. So these are clean rooms, which means that because the devices are so small, even a small piece of dirt will destroy the device, right? So that's why the air has to be very clean. That's why it's called clean room. So there are nanofabrication facilities in which we make these devices. In terms of, to give you an idea how small these are, if you think of a grain of rice, this would be 1,000,000 times smaller than that. So something which you cannot even see through your naked eye. So
gosh.
very, very tiny. They're subcellular in size, much smaller than size of even a single cell in our body. So they can actually live in between the cells without the cells even feeling that something is there. So yeah, very, very tiny.
Wow. So something that's that small, how do you power it, and how does it actually work once it gets inside the brain?
Yeah, so these devices have the characteristics that you can think of them as transducers. They basically convert these wireless electromagnetic fields into electric fields. And that is based on the electronic properties. We basically make layer by layer heterostructures and impart properties, electronic properties, that enable them to convert these electromagnetic fields into electrical power.
So you, the scientists, are applying an electromagnetic field?
Yeah, we design the device, such that it can do that functionality.
Okay.
Mhm.
And how does it work once it's inside the brain? How does it recognize those regions? What is it actually doing?
Yeah, so what it's actually doing is it is converting these electromagnetic fields into electrical fields, and it can then electrically stimulate the neurons. So our body, if you can think of this bioelectricity in our body, is a very fundamental phenomena. From thinking of the brain to beating of our heart to twitching of our muscles, bioelectricity is involved. And also it is involved in how cells communicate with each other. These are like tiny electric fields that are generated in the body between the cells, which is called bioelectricity. And if you can modulate and change this fundamental bioelectricity in the body, it is possible to treat diseases which, say, chemicals and drugs cannot treat. And how the neurons communicate with each other is also through these bioelectrical signals. And when diseases happen, this bioelectrical signaling is disrupted. So if you can correct these aberrant bioelectrical signals through these tiny devices, so it is possible to treat diseases. So this technology itself is a platform technology, so it can be used in multiple diseases. In our lab, we have already shown the applications for diseases like brain cancer, for Alzheimer's, chronic pain, and you're also exploring treating mental illnesses like PTSD, depression, and even it can be used for treating brain injury. The applications are limitless. And to give you a few examples of diseases that you have worked on and the impact you have seen. So, in the area of brain cancer, for example, think of glioblastoma. So, currently, there is no treatment for glioblastoma.
Okay.
Drugs do not work.
Okay.
Radiation do not work. And after diagnosis, the median lifetime of a period is 12 to 18 months.
Oh, man.
So this is horrible
Yeah.
because if someone is diagnosed with this disease, it's a death sentence because it's moving towards absolute death. There is no cure. Nothing helps. So our work has shown that in our animal work that we have done, that customized electrical stimulation can completely halt the growth of brain cancer.
Wow.
And this we have demonstrated taking tissues from patients who have died because chemotherapy, radiation therapy did not work.
Yeah.
So we have collaborations with hospitals from whom we get the tissue. And you have created animal models of brain cancer
Okay.
using those tissue. And in our animal studies, which are clinically relevant, we have shown that with our electrical stimulation, the tumor completely is halted. It does not grow any further. And this can increase the survival rate of patients to more than 85%.
Wow.
So this is very, very promising.
Yeah.
And we're thankful to the Sontag Foundation, who had actually funded our work and helped us to forge into this field of brain cancer. And there is another type of brain tumor. It's very, very horrible because it happens in children, small children, and below, like maybe 10 years old. And it again is a terminal cancer, no cure. Median life period is even smaller in this, like 8 to 12 months.
Oh, man.
So just imagine a parent, whose child would be diagnosed and there is like no hope.
Yeah.
There are also, our work, because it shows that it can treat brain cancers where chemotherapy and radiation does not work. It's very promising. Moreover, this brain cancer, which is called DIBG, this happens in a brain region where surgery is not even possible.
Oh.
It happens near brainstem, where it's a very sensitive brain region, so surgery is not possible. So it's just left alone when it is diagnosed. But since our tiny devices can go through the both body fluids and is able to go to the regions where surgery is not even possible, inaccessible regions in the brain which other technologies cannot access. So these devices are able to treat those kind of diseases. And this work has been supported by the Chat Tau Defeat DIBG Foundation. So our work is supported by parents of children who have lost their kids to this intractable disease. Yeah.
Wow. So you've started to touch on this already, but I'm curious why there is a need for surgery-free brain implants. What about our current approaches aren't cutting it, pun intended?
I think few things here. So the first one is the invasiveness of the surgery.
Yeah.
So you would need to create a hole in the skull and putting a centimeter scale probe in. Highly invasive. So most of the patients do not even want to go through this like very invasive procedure.
Yeah.
But many of these patients are not even eligible for a brain surgery, just because it cannot be performed on a sensitive patients, like
Okay.
Patients like small children or aged patients.
Okay.
Because surgery involves inherent risks of morbidity, mortality, risks of infection. Currently, these patients have this impossible choice: either choose a very risky brain surgery or live with the debilitating symptoms which drugs cannot fix.
Yeah.
So the invasiveness and the risk of surgery is one. Second is the hundreds and $1,000s of surgery-related cost. Brain surgery costs a lot.
Yeah.
That's the second thing. Third thing is that, as I mentioned, one example with DIBG, that there are regions in the brain which you cannot even surgically reach.
Yeah.
So there are sensitive regions where if you are to put an implant, that will cause more damages. So surgery is not even possible. So that's another scenario. And apart from that, there are diseases, and cancer can be one of the examples here as well, where there are small pathological sites where you cannot even image those pathological sites. So you do not know where to put the implant in. So they're so tiny, MRI cannot detect those.
I see.
So that's where the autonomous detection principle is really, really helpful, because our devices do not require external imaging.
Right.
They are able to detect the disease regions through biochemical sensing mechanism. So they are able to go to the regions of interest in the brain, the disease regions where you cannot even image those. So, if imaging is not possible, implantation is not possible. You do not know where to implant anything.
Right. Right.
So, and for example, again, taking the example of cancer. So, cancer, what happens is that there are like microscopic tumor sites that are there in the brain, which MRI cannot detect. So, even when a surgeon tries to surgically remove that tumor region, there are those diffuse microscopic sites which remains there,
Yeah.
which cannot be surgically removed. So that is another scenario where the ability to non-surgically go there and autonomous detection helps in. But yet another advantage is that surgically you cannot put in a lot of electrodes inside the brain.
Yeah.
It would seem like an acupuncture of the brain. But because our devices are very, very tiny, even if you have to target multiple regions of the brain, these devices can go there. Because most of the diseases, they are not confined to a single node inside the brain. There are multiple regions in the brain that needs to be treated. These devices have the capability to simultaneously go to multiple regions in the brain and treat those, where you cannot put invasive electrodes in multiple regions. Normally, it's just done like 2 electrodes at most. Because once you put the electrodes, a large region of the brain gets destroyed around the electrode. So that's also another promising, exciting results we have shown, that once these electronic devices are in the brain, the cells do not even feel that something is there.
Because
So
they're subcellular.
yeah, because they're subcellular. So these electronic devices, they co-live and coexist with brain cells. And the brain cells do not even feel that something is there, and they feel like they're completely healthy. And this we have actually shown our brain samples to both certified pathologists, and they gave the same score to the brain with our devices and without the devices. They said that the brain is not even feeling that something external is there, because this is so tiny.
Wow.
And compare that when you put an electrodes, you see, like millions of neurons getting destroyed around it.
Wow.
Yeah.
So it sounds like this is so much less invasive than a surgical implant. How are the devices actually getting delivered to a person, if not, cutting a hole in the head and sticking them in?
Yeah, so the devices, how they go to the brain region is basically traveling through the body fluids.
Okay.
So they can be introduced, for example, into the blood stream.
Okay.
So like a simple injection, or through the cerebrospinal fluid, or even through the intranasally as well.
Wow.
So you need to introduce them like very, very non-invasively, similar to how a person with diabetes would take a simple injection by themselves. So once you introduce through the peripheral body fluids, the devices, because of their biochemical recognition capabilities, are able to go to the target region of interest and self-implant there. Yeah.
And how do they know to get to the brain? How do they know if these regions that we're talking about are so small? How do they do this biochemical sensing and do the things that they need to do?
Yeah, so that property comes from this biomolecular machinery that we have. So they have specific ion channels and sensing mechanisms in them. So they check and they actually go through the chemical gradients. So these are not passive antibodies which are just going everywhere and seeing something and getting attached.
Right.
But they can sense the chemical gradients, so they're actively going to the region of interest.
Okay. So I'm curious about the applications of your work beyond the realm of diseases in the brain. Do you see a place for these implants one day in healthy individuals, and in what contexts?
Yeah. So yeah, as I was mentioning, we have a lot of work done in brain cancer, but also in Alzheimer's, we have shown that if you can, in Alzheimer's disease, that if you can provide very customized and precise electrical stimulation, you can actually stop this neurodegeneration from spreading to other brain regions.
Wow.
So, if you can apply this technology early on, it would be possible to even prevent the development of Alzheimer's disease or other neurodegenerative diseases. And prevention is, of course, always better than cure. So, that is another application. But you also have applications in other diseases such as chronic pain. So, our first focus is to treat diseases,
Yeah.
and the idea and the we envision that we will be able to reduce human suffering. But in the future, it is possible to go beyond diseases and also use them in healthy individuals. The reason is because this is a non-invasive procedure, completely non-surgical. So even healthy individuals can use it. So just like wearables for all, we can also have implantables even for all.
Wow.
And some examples of things it can do. For example, you can think of increasing brain capabilities, improving cognition by precise neuromodulation. As I mentioned, brain computation happens through electrical signaling, and we can modulate them precisely. And you can also think of increasing the neural density, increasing the number of neurons we have in the brain with these synthetic electronic neurons.
Wow.
So these devices can work as synthetic electronic neurons. And you have already shown that these electronic devices co-live and coexist with brain cells without damaging them. So that part is already shown.
Yeah.
So they can get distributed around neurons. And the electric power they can harness right now, and at the device scale of even if it is million times smaller than a grain of rice, it is possible to integrate like multiple sensors and transistors into this device and build nanoelectronic circuits. So they can do work, they can have functions beyond just electrical stimulation, but also sensing and analysis of the data in situ itself. So, and down the line, even creating artificial neurons with those.
Wow.
Yeah.
So far, you've done your work in animals and animal models. How long before we would see this being used in humans, and what needs to happen to get to that point?
Yeah, that is a very good question. So, yeah, this technology we developed in our lab at MIT over the last 7 years.
Okay.
And we have now robustly demonstrated this in even like clinical studies with animals.
Okay.
And the next step is really for us to scale it up and take it to the clinic so that it can help humans. And that is one of the reasons that I recently launched a company to take this work forward. And the idea is that the company would accelerate the research and take this work to humans.
Okay.
And our estimated period is around 3 years where we are able to take this to clinic.
Okay.
And the company, actually, I have named Kahira Technologies.
Oh.
So Kahira in Irish, it means a warrior.
Okay.
Specifically, it means a female warrior.
Oh.
So I chose this name because of our vision of defeating intractable diseases and overcoming biological limitations.
I love that. That's so cool. So your work has been likened to science fiction. And in fact, you yourself even said that sometimes science can be stranger than science fiction. What sort of feedback have you received from the scientific community and beyond about the work that you're doing with these non-surgical brain implants?
Yeah, the response we have received is very, very interesting. And it's it flipped from the initial response to the, response later on.
Okay.
So initially, when we started doing the work in the first 1 or 2 years, And then I was, of course, as a professor at MIT, I was writing grants to get this work funded. For the first 2 years, more than 35 grants I wrote got rejected in a row.
Oh, no.
Because this was considered an impossible technology. All the reviewers mentioned that this is very impactful. We can clearly see the impact if it can be made possible. But this just seems like it can never be done, and it's not a moonshot. It's a Mars shot.
Okay.
So it's probably not even useful to fund this work because it's probably not going to go anywhere.
Wow.
But the response completely flipped when we started doing experiments in our lab and had initial data already. Started to show that early evidence that these are possible. So we are able to show that we can actually implant devices, electronic devices, in a targeted brain region with precision of few micrometers. We showed evidence that this can cross the blood-brain barrier. We can electrically stimulate, reach regions where other devices or other implants cannot go. After that, we got a huge positive response, and we got a lot of accolades and awards for that. So we got the NIH Director's New Innovator Award for this technology. This is one of the highest honors that NIH provides to early career scientists. And we not only got that award, we got the highest score that NIH provides to any project. So they score projects from 10 to 90.
Okay.
And you get 10 when basically all the 50 committee members and experts in the field, they, along different criteria, give you 1. Like that's the best score. And then they take an average and multiply by 10, something like that, to come up with that perfect score. Actually, as a initial, as a scientist writing NIH grants for a few times, I didn't know that the 10 was the best score.
Okay.
I thought it is 1 to 100, and 1 would be the best score.
Okay.
10 is still a good score. So I looked it up, and when I got the score, and then I said, 10 is actually the best score. That it's called a perfect score.
Wow.
And very few people in the world gets this perfect score, because if any of the reviewer thought that instead of 1, I'll give you a 2 is lacking in something, then you do not hit the perfect 10, right?
Right.
So I was super excited about that all the 40 to 50 reviewers that would be forming the committee, they gave each of them in different 6 to 7 criteria about impact, innovation. They all gave us 1, and I often tell my students that there's nothing like a perfect thing, right? Nothing is perfect in the world. So the reason the reviewers gave us a perfect score is not because this technology is perfect, but it shows the excitement and the impact it can have. And the NIH official statement actually mentioned that this technology seemed like something from a science fiction novel.
Yeah.
Yeah, and then the statement also mentioned, but the data shows that this is possible. And also the NIH statement, the official statement mentioned that this technology, it can herald a new era.
Wow.
So that's what that was one of the first grants that we received. We had also received a lot of other accolades. For example, we received the Mind Prize. We were one of the 7 researchers nationwide to get this award. And this was highlighted in New York Times. And this was for treating Alzheimer's disease at an early stage, so that we can prevent even the formation and development of the disease. And the Sontag Foundation New Innovator Award, we got it for treatment of glioblastoma. There we were one of the 3 researchers nationwide. And we were actually the only group from MIT to get this award in the last decade.
Wow.
So this is one of the highest honors in the field of brain cancer.
Wow.
Specifically glioblastoma. And we also got the Chat-Tau Defeat DeepG New Investigator Award. And this is for treating DeepG, which happens in children. And because of the potential of this work to go to brain regions where no other technology can reach. So we had been getting a lot of positive response even before the paper has been published.
Yeah. You had spent so long submitting these grants and receiving no's and just hearing that it was this outlandish idea. What did it feel like when you finally got the yes, when you were finally told that this could work?
Yeah, so initially when I was getting the no's, it was, of course, a very, very challenging time for the, first few years. And I remember those times I was writing so many grants, I would often sleep like alternate nights to meet the deadlines. So every month I was skipping several days of sleep just to meet the deadlines. And this was after the fact. I was already working 110 hours a week, so even that was not enough, that I would have to skip sleep to make sure we can meet all these deadlines. And so this continued for a few years, and at some point I was thinking that we would have to show all the results. It was clear because it is so different. Until and unless we show the results, we would probably not get attention. And then when we got it, it was almost like unbelievable. And we got like such a huge response. It was not that we just got the grant, but as I said, you got the perfect score of 10. So that was like unbelievable because, as I said, I didn't even know that 10 was the perfect score. I was thinking 1 is the perfect score, so 10 is already good enough, so get in. So it was, I was overjoyed. I was very much excited that finally, that people are seeing that this is possible. So it was, I think it's even difficult to express in words the feelings that I had that time, because it was like almost no to a like perfect yes. And it was amazing feeling.
That is really, truly amazing. So clearly this is, a huge, impactful idea and developing technology. Where and how did you even come up with the idea to create non-surgical brain implants?
My background is actually in nanoelectronics and applied physics. So I was building tiny nanoelectronic devices throughout my bachelor's and my PhD work. Initially, my work had very little to do with biology. So I developed a quantum mechanical transistor where I showed it can overcome the fundamental limits in power of existing electronic devices. And while working on these low power electronic devices, I realized that brain is the lowest power computer ever. So that got me interested in the brain. And so when I was like finishing up my PhD, I decided to do a very steep transition from the field of electronic devices and nanoelectronics to the field of neuroscience and neurotechnology. And then I started learning like a bachelor student again, like about the brain, about neuroscience and neurotechnology. And then I realized the gaps in the field of neuroengineering and neurotechnology that is there. And I also realized that because of my background in nanoelectronics and applied physics, I could bring things from that field of nanoelectronics to this field of neuroscience and neurotechnology. And because I was involved in building cutting edge, very, very tiny nanoelectronic devices, those I was building for that would go into the computer for energy efficient computing. But now this could have applications in the brain. And I also became aware of the surgical risks and that there are regions in the brain you cannot even reach surgically. And I realized that if we can make these devices very, very tiny that can travel through the body fluids, you can overcome those challenges. So it was kind of a fusion of my previous background in nanoelectronics, and then my foray into the field of biology. That I had ideas and concepts from both the fields that I could combine. So when I started my own lab at MIT, I decided to fuse the fields of nanoelectronics, applied physics with biology. And I believe that if you can combine dissimilar fields, you can build something very, very unique and impactful. Yeah.
I love the idea that you were building nanoelectronics for computers and then realized the brain is a computer and combined them in that way. I'm curious what got you interested in nanoelectronics in the first place, and I guess even farther back than that, what got you interested in science in the first place? Where did you find your spark?
Yeah, I think the spark for science was probably from the very childhood, because I used to see my father build scientific projects at home.
Okay.
So, for example,
what kind of projects?
he built a washing machine which can work without the need for any electricity.
Whoa.
Yeah, he built a system of pulleys where you can bring up very heavy objects onto the roof with very little effort. And where our home is in India, it is not a main city area, so the cars will not go all the way up to our home. So it's a little bit distance. So if you have to carry heavy objects, or if someone got sick and he needs to go to the hospital, so for that, my father, in fact, even built a whole vehicle by himself. So if you have to carry someone in the small alleys, that can be done. So I grew up seeing him, doing this scientific project. So that got me interested in science and specifically engineering, because engineering you can actually build things for the good of humanity. So that got me interested in science and engineering. And specifically physics, applied physics was very, very exciting for me. Initially, actually, I did not like biology that much.
Okay.
Part of the reason was biology was often like things, the way it was taught in school, you had to remember a lot of things like the body organs and their functions. But biology is very, very exciting and interesting once you go deep inside it. But I did not see that part when I was, when I was in high school and all those things. So physics was very, very exciting. So that kind of got me into nanoelectronics. Yeah.
Nice. Deblina, thank you so much for being here and talking about this with me. This has been really amazing.
Yeah, thank you so much for having me here.